Hi, in this video we are going to talk about hemoglobin and its oxygen binding kinetics. So we know that hemoglobin is a key molecule or the key oxygen transporter molecule situated in our red blood cells. Now hemoglobin is a large protein with at least 4 subunits, and each of these subunits are composed of heme moiety, which is a non-protein structure. It's a protoporphyrin ring, but along with it, it has the globin protein. And each of these subunit weighs around 65 kilodalton in molecular weight. Now, here is hemoglobin. So there is the globin part, and there is a heme part. Now, heme part is the particular moiety which binds to the oxygen. So when we talk about hemoglobin and it is binding with the oxygen, we have to understand how oxygen is binding to the ferrous uh, in the hemoglobin in the heme moiety. So the hemoglobin consists of globin. Now globin is a protein, so globin is encoded by alpha and beta globin genes located in chromosome number 16 and chromosome number 11. So then we need to understand where does the heme come from and how the heme biosynthesis takes place. So heme biosynthesis takes place from an intermediate of Krebs cycle known as succinyl-CoA. Now the succinyl-CoA and the amino acid glycine is together used to form delta aminolevulinic acid, which gets eventually converted to heme, right? Now this heme can bind to the oxygen. But the hemoglobin, the globin chain for the hemoglobin might be very different for different type of hemoglobin. And it turns out that there are several different types of hemoglobin present in human body. But their abundance are pretty low. For example, HbA2 or HbF, their abundance is pretty low. HbA1, which is composed of 2 alpha residues and 2 beta residues, 2 alpha globin residues and 2 beta globin residues are having the highest abundance. In case of fetal situation, the hemoglobin is different. It's known as embryonic or fetal hemoglobin or HbA. In that case, the subunits are alpha subunits and the epsilon subunits. And fetal hemoglobin has total different affinity towards oxygen than the adult hemoglobin. So that would be topic of another video. But in this video, what I'm going to talk about is the structure and the function of these hemoglobin is pretty much conserved throughout several species of the animal kingdom. So it's evolutionary conserved. So we obtain oxygen from the environment. So when we breathe, oxygen comes in our lungs, and inside our alveoli, which are the air-filled sacs, it is surrounded with blood vessels. So in the blood vessels, there is exchange of oxygen and carbon dioxide, but oxygen cannot dissolve properly in the blood because its solubility is very low. So it binds to the oxygen carrier protein, which is the hemoglobin. And in this video, we're going to learn about the binding kinetics. So let's look at the structure from a side view. So here is the side view of the protoporphyrin ring in the hemoglobin. So you can see the ring is pretty much in a non-planar wedge shape when the oxygen is not bound. But when the oxygen binds, the conformation of the ring changes. The ring becomes planar in oxygen bound state. And you can see the nearby histidine residues are bound to this oxygen with several non-covalent interactions. And this conformational change would increase the probability of the nearby site for oxygen binding. And this kind of binding method is known as cooperativity. And this is an example of a hemoglobin which is deoxygenated right now. None of these subunits are bound to oxygen at this situation. That's why it's looking pretty much purple. And that's why our veins look purple or bluish. Now, then it binds to one molecule of oxygen and one molecule binds to the heme group. And that increases the probability of binding in the next subunit. So in this fashion, in a progressive and sequential fashion, the binding is facilitated by the binding of the oxygen in the first subunit. And this kind of binding method is known as cooperative binding. But how do we understand this binding in a mathematical format? In order to understand that, we have to understand Hill equation. which is a mathematical format which would allow us to understand the binding kinetics. So let's look at it. Imagine we have a protein which we are saying P, and it's a multi-subunit protein, so it can bind with several ligands. In this case, the ligands are known as L, and there are n number of ligands. So it would create a protein ligand complex like this, and we are saying the association constant or this equilibrium constant is Ka. So we can define Ka and we can derive the expression for Ka, and the Ka's expression would pretty much look like this, right? Then what we can do, we can express the equation in terms of a binding fraction or saturation fraction. Saturation fraction simply means binding sites occupied divided by total number of binding sites. So this pretty much looks like this particular mathematical equation, PLN divided by PL plus LN. Now substituting the value of PLN, which we derived from the K association constants expression, we can get an expression like this. And a little bit juggling around the terms and taking common, we can derive that theta or the binding fraction is ln, L to the power n divided by L to the power n plus 1 by Ka. 1 by Ka term is actually dissociation constant, because dissociation constant is just opposite of the association constant. So we can modify the phrase and write it like this. Now, if we do the math a little bit more and take log in the both sides, before taking log, we need to rearrange it on the both the sides. So theta by 1 minus theta looks like ln divided by kd. So now we take a log in both the sides and we end up with this kind of expression. And this expression is basically the Hill equation, and the n is here the Hill coefficient. In a graph, it would look like something look like this. So here in the y-axis, you have log theta by 1 minus theta, which is also known as binding fraction or saturation, percent saturation, and the ligand concentration is given in the log of ligand concentration is given in the x-axis. So you can think, if we talk about hemoglobin, so it would be partial pressure in case of the concentration. So the curve looks like this. So at lower affinity state, the n or the Hill coefficient is 1, but the Hill coefficient is high for the higher affinity state. Oops, here the higher affinity is written as lower affinity. That's a clean mistake, sorry for that. But anyway, so let's move on to the next curve to understand how the partial pressure of oxygen changes and how that affects the binding of oxygen to the hemoglobin. So here, this log theta by 1 minus theta term, we can write it by percent saturation of hemoglobin, and the log L term we can write it as like a partial pressure of oxygen in this alternate term. Now let's look at this portion of the graph. So here the curve has flattened and it's pretty much reached a plateau. In this situation, if we increase the partial pressure of oxygen, we can see the y-axis. There is not much change in the percent saturation. But look at the ascending phase of the curve. In that ascending phase, if we change the partial pressure of oxygen even a little bit, like this much, then there is a huge change in the percent saturation. That means the binding kinetics changes even you change a little bit amount of the partial pressure of the oxygen. So this particular ascending phase, in this particular ascending phase, the change in partial pressure of oxygen can change the oxygen binding kinetics, where at higher concentration or higher saturation fraction, changing partial pressure does not make any sense. Let us understand how. So at the ascending phase, we can see the phenomena of cooperativity. So each of these oxygen molecule binding to one subunit would allow the other subunit more likely to bind to the oxygen, and this cooperative nature of binding allows the increase in percent saturation in the small increase of partial pressure of oxygen. But at very high level of saturation, all the sites which can bind to the oxygen are occupied. So even if you change the partial pressure of oxygen, there are not even enough sites where oxygen can bind. And that is how we need to understand the oxygen dissociation curve. So this is the pretty much initial video about the oxygen dissociation curve, where we touched upon the Hill coefficient and derived it. But there are several physiological factors such as O2 concentration, CO2 concentration, pH, temperature, and presence of other intermediate such as 2,3 BPG can shift this curve either towards right or towards the left. And each of that has a particular physiological and clinical significance. About that, we're going to look at in a different video. So stay tuned till that next video. I hope this video was useful. If this video was useful, give it a quick thumbs up. Don't forget to like, share, and subscribe. Just to let you know that I'm also present in Unacademy. You can have a one-on-one session in Unacademy with me. And with that, you have to join the Plus subscription, which is pretty cheap. And you can use my code EPI10 to get a 10% discount. So stay home, guys, and stay safe. Thank you.